Hello everyone, and welcome to our next presentation entitled Generation and Application of 3D Organoid Cell Culture Models Using Human-Induced Pluripotent Stem Cells for Drug Discovery, which will be presented by Ryan Hicks, Associate Director of the STEM and Primary Cell Group at AstraZeneca. I'm Roland Others from Thermo Fisher Scientific, and I will be your moderator for today's presentation. Before we begin, I'd like to remind everyone that this is an interactive event. So we encourage you to participate by submitting any questions you may have at any time during this presentation. Just click on the Ask a Question box located on the far left of your screen and type your question into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you are having trouble seeing or hearing the presentation, please use the Ask a Question box to let us know of any technical difficulties. With that, I would now like to introduce you to Ryan Hicks. Ryan is currently Associate Director of the STEM and Primary Cell Group at AstraZeneca. Ryan's group is responsible for the generation, engineering, and differentiation of iPS cells, adult stem cells, and primary cells for assay development, target validation, hit finding, hit to lead optimization, and phenotypic screening activities. Cellular models are generated in 2D and 3D culture systems, and part of the model development work includes using precise genome modification techniques to introduce gene knockouts, knock-in mutations, or endogenous reporters into the iPS cells. The group's work primarily focuses on AstraZeneca's cardiovascular, metabolic, renal, and respiratory disease areas. Ryan joined AstraZeneca in 2001 and has been based in Sweden for the past 8 years. Prior to working in Sweden, Ryan worked as the Associate Director of the Cell and Protein Reagents Group at AstraZeneca in Alderley Park, UK, with responsibilities within the cell and protein disciplines. Ryan received his degree in medical biochemistry from the University of Birmingham, UK, and his PhD in biochemistry and molecular biology from the University of Leeds, UK, in 2001. And with that, I would now like to turn the presentation over to Ryan.
Okay, thank you, Roland. Thank you very much for the introduction. As you stated, my name is Ryan Hicks, and I would like to talk today about the generation and application of 3D organoid cell culture models using human-induced pluripotent stem cells for drug discovery. The way I'd like to do this is to first off tell you a little bit about AstraZeneca and about the STEM and primary cell team that I lead, and also give you some background on the types of things that our team does. I will then move on to talk about 3D organoids, and I will do this by using our 3D kidney organoid model as an example to highlight the work we've done in the organoid space. And this will include why we are generating 3D kidney organoids, how we are generating them and validating them, and also how we use them in a drug discovery setting. So AstraZeneca is a global biopharmaceutical company with around 60,000 employees, and we have 3 main focus areas. And these are cardiovascular, renal, and metabolic, oncology, and respiratory, inflammation, and autoimmunity. We also have opportunistic areas, and these include infection and neuroscience. And each of our focus areas is driven by our Innovative Medicine Biotech Units, or IMEDs. And these IMEDs are supported by centralized departments, including Discovery, Safety, Metabolism, and Discovery Sciences, of which I am a part. Discovery Sciences is a central group that provides state-of-the-art support for hit finding, lead optimization, target validation, and this is for all therapeutic research areas across AstraZeneca. And we do this through partnering with each of the therapeutic areas on the projects that they have. Within Discovery Sciences, we have a number of different centralized groups, and examples include sample management that handle our compound collection, high throughput screening, protein structure and biophysics, and several others. The department within which I belong is called Discovery Biology, and our mission is to be at the innovating front of science and technology to deliver reagents, assays, and disease models to discover new targets and medicines. Our Discover Biology group is located across 4 sites, 2 of these in the UK, 1 in Sweden, and 1 in the US. The stem and primary cell group, which I lead, is based in Gothenburg and is a global group supporting areas across AstraZeneca. And we focus on generating cells and cellular models that fit with the focus areas of AstraZeneca. So these include hepatic, lung, immune, kidney, pancreas, cardiac, adipose, and neuro. And in generating these models, we use many supporting technologies, and some of these include precise genome editing, and we use this to genetically manipulate our cells, and these are technologies like CRISPR. We also culture our cells in a variety of formats, and these can range from 2D through 3D, microphysiological systems, and humanized mini organs. And by that I mean tissues that have been decellularized and then recellularized with stem cells. We also use techniques such as high content imaging and next generation sequencing. And this is in support of both the quality control and the validation of models, as well as looking at their function for screening and application in a drug discovery setting. It's also very important to the group to have an external presence, and the way we do that is through having postdocs within our lab and students, including PhD students and master students, and we also have a number of collaborations. And a lot of this is pointing towards publications and presenting at external conferences. In this slide, I'd like to give some examples of how our team provides stem cells in drug discovery. And these models are all iPS derived, some of which have been manipulated using precise genome editing and are grown in single cell, 2D and 3D formats. So starting in the top left-hand corner in the blue box, we had a project team that was looking at an anti-inflammatory response in macrophages. And this was mediated through salt-induced kinase, or SEEK. Now, SEEK comes in 3 isoforms. So the team approached us as they wanted to know which of these isoforms was driving their desired phenotype. So what our team did is that first we looked to generate a differentiation protocol to take our iPS cells through down past monocytes to macrophages. So once we developed a protocol to generate macrophages from our iPS cells, we then applied precise genome editing using CRISPR to modify our iPS cells. And what we did was to systematically make catalytically dead versions of each of the SEEK isoforms in different cell lines, so that the cell would express each SEEK isoform, but the isoform itself would be catalytically inactive. We then took each of these isoforms together with the parental cell line and differentiated them down to macrophages, and then applied them in the project team assay, and then looked at their response to different compounds. And from this, we could identify which of the isoforms was responsible for driving the phenotype. And this enabled the project team to direct their chemistry against the right isoform of SEEK to be able to drive new medicines against the anti-inflammatory response. The second example I'd like to talk about is in the bottom left-hand corner in the pink box, and this is in the cardiac space. And here the project team was interested in finding agents that could specifically induce the proliferation of cardiac progenitor cells. So again, taking iPS cells, we generated cardiac progenitor cells and applied them into an assay to be able to measure the proliferation rates. So we wanted to measure the basal proliferation rate, and that in the graph is shown in blue to show the proliferation rate of the cells in normal conditions, and then in the presence of a positive control, in this case FGF, to see the maximal proliferation rate. We then ran a screen using small molecules to see which small molecules we could find that would induce proliferation of our iPS-derived cardiac cells, and then counter-screen this against other cellular models to try and find compounds that were specific for proliferation of cardiac cells. The third example is in the pancreatic space. So we have an interest in diabetes, and so protocols that can help us generate pancreatic beta cells are very interesting to us. A useful tool to aid the development of protocols, as well as having online QC and supporting in screening of compounds, is to have an endogenous reporter tag in that cell line. In this case, for our pancreatic cells, we genetically modified our iPS cells to contain a GFP reporter downstream of the NKX6.1 gene, an important factor that you should see coming up along the differentiation route of iPS cells towards pancreatic beta cells. Here you can see in the images on the top row the differentiation of the parental line down the pancreatic lineage and expression of NKX6.1 as done by staining and imaging, which we need to terminate the culture and image and fix. Whereas in the bottom panel, you can see with the GFP reporter line, you can see GFP expression together with NKX6.1, showing that the reporter can give you a live readout of NKX6.1 expression. And I will talk more about the generation of endogenous reporters later in the presentation. Finally, on this slide, you can see a kidney model. And here we have generated 3D kidney organoids using a reporter line and applied this to a number of projects. And it is these organoids that I'll focus on in this presentation. So in this slide, I just wanted to give you a bit of background on what we wanted to use these organoids for and what features we were looking for. So in the kidney space, we've worked on a variety of kidney cell types, generating kidney cells from iPS in 2D formats, 2D co-cultures, and also in 3D formats. And it's this 3D format that I shall talk about today. These organoids we have applied in a multitude of different settings, and this includes disease modeling. And here we took cells in normal and disease states, using either culture conditions to get the disease state, or using precise genome editing to genetically modify the cells to see a disease phenotype. We also use them for target identification purposes, supporting activities like phenotypic screening of these organoids to help identify new targets. We also apply our organoids to target validation activities. So taking targets that have either been identified internally or externally, for example, from the literature, and then validating these targets. And by validating in this context, I mean to confirm that the target that has been identified is responsible for the disease state, or that if you modulate that target, it's responsible for causing a disease state or returning that cell back to a normal state. We also use them in a safety and metabolism setting as well to look at the profile of molecules that we are developing. So for the phenotypes that we look at, I've listed just a few of the features that we might look for, and these include cell-specific uptake or target engagement, or it could be things like clearance of a compound or its toxic effect. Some key requirements of the model are that it must be translatable. So the reading we get in our in vitro setting translates through to the in vivo setting, and ultimately it needs to translate to the patient setting, so that the readout that we are seeing in the in vitro model is what we would see in the patient setting, so we have a predictive model. It also needs to be reproducible. We need a robust technique that we avoid false positives and false negatives, and we can trust the data time after time. All of this is for the ultimate aim of driving our drug discovery projects. So now I'd like to talk a little bit about what has driven us and driven the need for us to generate these in vitro 3D organoid models. If I take it at a top level for looking at chronic kidney disease, there is a very high prevalence. It's estimated that over 10% of the world population is affected by chronic kidney disease. And indeed, the mortality rates are seen to increase. So between 1990 and 2013, we have seen a doubling in mortality rates. And if you look within the UK, chronic kidney disease kills more people than breast cancer, lung, colon, and skin cancer combined. In terms of treatments, there are very limited options for chronic kidney disease treatment, and so there is a high unmet medical need to advance new medicines in the field of chronic kidney disease, and for that we need in vitro models to help discover those medicines. So looking more specifically about why we need an in vitro 3D kidney model, as we've talked about, there's some general needs in drug discovery for models in terms of they need to be translatable, rapid, and reproducible. And also, I've talked about some of the activities that need these. It's target identification and target validation, and use in screening, as well as safety applications. So what is driving the need for 3D models? So what we're seeing is that there are 2D models in place today, but many of these show a lack of translatability, where the response you see in these 2D models does not recapitulate in the patient setting or is not predictive of other models. There can be a number of reasons of why these two models don't translate well. Some of these include a lack of expression of key genes and proteins that you expect to see in the model or in the patient setting that do not come up. These models are often generated with immortalized cell lines, and so if you want to see a gene that's not there, you have to overexpress your target. And this is not ideal from a biology point of view, nor is it ideal from a model point of view that you have to generate a number of different cells to generate your different models. These models can also be derived from primary cells. This does give some advantages, but also the donor cells from primary cells from patients, you do see donor-to-donor variability. Their supply can be limited, and they can also differentiate really rapidly once they're out of the patient, and so do not resemble how they looked when they were in the in vivo setting. A final point is that, by definition, a 2D model doesn't contain the 3D structures that you see within the kidney. And so if you're wanting to look at some structural components, then that's not possible, and you would need a 3D in vitro model for that. Stem cells do have a number of limitations, but they also offer a route to generating improved models of this in a 3D setting. This slide is just to highlight what our 3D kidney organoids look like, so you have that in your mind when I go through this presentation. So in the middle of this slide, you can see a diagram, a picture, of one of our organoids where it has been stained. So in the light blue staining, you can see WT1, which was a surrogate marker for podocytes. You can also see in red LTL staining, which is a surrogate marker for our proximal tubule cells. And in green, you can see CD31 as a surrogate marker for endothelial cells. So in our organoid, we are seeing the assembly of and clustering of different cell types and cell types that we would want to be seeing in our kidney organoid. So I'd now like to move on and tell you a bit about how we built our model. And as I touched on earlier, a great advantage to model building is to have a reporter cell line. And this can give you live information, both on how your iPS cell line is progressing down the lineage for development of protocols and also for QC purposes. And so the first step in what we did was to generate a fluorescent reporter line. As we talked about, we did this using precise genome editing, and we're fortunate within AstraZeneca to have a very strong toolbox of precise genome editing capability. And we have a number of things within that toolbox that we apply to our stem cells for different projects. And these include the knockout of genes to remove the expression, and this can be simple straight knockouts or conditional knockouts that are inducible. We also do knock-ins, whereby we insert DNA sequences, for example, to create disease mutations within the iPS line. And this we did to make, for example, the functional dead mutant that I talked about earlier for Seq. What I'll focus on more is about how we've used it to introduce endogenous reporter tags, and that will come up in later slides. And we also have CRISPR forms that modulate expression, so CRISPR I and A technology, whereby we can use CRISPR to either inhibit the expression of a gene or to activate its expression of a gene. So when considering the markers we wanted to highlight to generate our kidney cell types of interest, we thought about what genes along the route, the generation towards our kidney-specific cells did we want to see. So starting in the top left-hand corner, the markers we would like to see in our undifferentiated stem cell, then going down the lineage towards kidney precursor cells, and then ultimately to the different cell types we would want to see in the kidney, and also genes we did not want to see expressed if they were going down the incorrect route. And this diagram shows that flow. And the first point we wanted to see a marker was at the kidney progenitor stage. And here we chose Hicks2 as our reporter. So what we wanted to do here was to introduce an endogenous fluorescent protein downstream of the Hicks2 gene, such that when Hicks2 was expressed, we could also see the expression of a fluorescent protein and the cell would change color. In addition to a single reporter, we also wished to make some double reporter cells so that we could see the tail change color again as it matured down towards our cell type of interest, in this case a podocyte as indicated by the star. And for this we chose nephrin. So again, what we did is we wanted to insert a different fluorescent reporter downstream of nephrin so that when this second gene expressed, a different fluorescent marker would also express and the cells would change color again. And these reporter lines would help us to define and refine our differentiation protocol, as well as giving a good QC to see how well our organoid generation was progressing. On the technical side of exactly how we built these lines, I've just added this slide to show the CRISPR system, which I believe most people are familiar with. But in brief, you use the Cas9 endonuclease to cut your DNA. And if you also add the presence of some donor DNA, you can get that to integrate specifically at a site and introduce that new DNA. On the right-hand side, I've given a schematic of that donor DNA. And what you can see is it's composed of flanking regions on either side, showing homology to the area in where we wanted to insert it. We also have a 2A site, and that helps to have the mRNA cleave into two parts when you're expressing the genes. And this is important to us because we want to see Hicks2 expression and also GFP expression, but we do not want the two fused together because we don't want to interfere with the Hicks2 expression. So that was why we introduced the 2A site. Then we have the fluorescent reporter. And following this, we have an antibiotic resistance cassette flanked by LOX-P sites. So this serves the function that when we're getting integration, we can use the antibiotics resistance to help select the clones with integration. And once we've selected our clones, we can use Cre recombinase to remove that cassette and avoid any unwanted side effects of having the antibiotic cassette in our cell line of interest. We did a similar thing with nephrin, and we did this in using multiple different reporters in single and dual reporter lines to give us a suite of cells that we could use to monitor our differentiation. When generating our reporter cell lines, we want to make sure that they are validated. And we looked at a few different areas here. So looking at their pluripotency, so differentiating the line down into the different lineages to make sure it is still pluripotent, that it can differentiate into the different germ layers. We also look for expression in the iPS cell of markers that show it is in still a stem cell state, and we can do this by imaging and also flow analysis. We also perform quite a bit of sequence analysis to ensure that the integration of our cassette was accurate, was faithful, and that we didn't see any off-target cutting from CRISPR or off-target integration of the cassettes. Genomic stability is also very important to ensure that the cell line has maintained its carrier type throughout the cloning process and also maintains carrier type throughout its culture. A further form of validation was to see that when we are differentiating our reporter lines down the kidney lineage towards podocytes, that we could see the co-expression of the reporter gene of interest together with a fluorescent reporter as well. So what we did was to take our IPS reporter lines and differentiate them using the protocols to hand and monitor by qPCR and by imaging the expression of our desired target proteins. And on the right-hand side, you can see a graph of one such measure where we have looked at mRNA expression levels of our 6-2 and see how that correlates with GFP expression levels monitored by flow cytometry. And we can see that the two are correlating nicely. So we were happy that we had generated reporter lines that were working nicely in our hands. Okay, so just to give you a few details about the protocol that we applied to differentiate our IPS reporter lines down the kidney lineage towards our 3D organoids. So we start with an initial 2D phase, culturing the cells for 10 days in 2D, after which time we harvest at day 10, and then we seed these cells into agar-well plates. And we like to use agar-well plates as this gives us a very precise control both on the cell number within the organoid and so the size that organoid will become. And we keep cells in this agar-well plate for 2 days to let them form these organoids, after which we harvest the organoids from the plate and then we grow them in suspension for at least 12 days to mature to the 3D organoids. We have several QC steps along the way to ensure we're getting reproducible results. And a great QC, as we said, is to use our reporter lines. So in the bottom right-hand corner of this slide, you can see images of 2 such reporter lines. The one on the left-hand side is showing a reporter of nephrin, which is a red reporter, and you can see expression of our red reporter. And the bottom right-hand is a second reporter line whereby nephrin is followed by a green fluorescent protein, and then you can see expression of GFP in our organoids. What you can also see is the expression is located in certain areas within the organoid, and I will come to this later on. This slide is just to quickly show some further QC using electron microscopy of our organoids, and in that you can see features that you would like to see within the kidney appearing in our organoid. So here with on panel A with transmission electron microscopy, you can see images of day 35 organoids showing features you would like to see, such as tight junctions, and also maybe some primitive foot processes from podocytes. And in the lower panel, a scanning electron microscopy images of the surface of the organoid, again when you can see features that you could see perhaps look or perhaps resemble slit diaphragms of the glomerulus. Okay, so I've given you some details of how we made the organoids, and I want to move on just to tell you a little bit more about that, but more so about the QC of these organoids and what things we consider when we're generating them to be able to apply them to a drug discovery setting. So we have published our protocols, and you can find this in the publication shown on screen. And in here, you'll find details on the protocol, and you'll also find further information on things like single-cell next-generation sequencing that we performed, and also further imaging analysis of these organoids. So when generating advanced models and thinking about applying them to a drug discovery setting, we need to keep some key components in mind, and I've listed these on the circle on the left-hand side. So just to touch on a few of these, thinking about culture format, thinking about how we grow our cells. Are they growing in suspension, or are they growing adherently? Are they as individual cells, or are they as clusters? And can these be adapted to different formats? And this is to support their growth and development, but also make sure that they are amenable to the technologies we're using to assay them. We need to consider the scale at which we can generate these cells. So to what extent can we scale up or scale out our methods to keep to keep the method faithful, but also to keep the cell numbers and reach the cell numbers needed for the screen that we would like to do. We have to have a quality model, and by this I mean, does the readout that we're reading in the in vitro assay, how well can we measure this? And does this readout, does this measure give a good surrogate measure for the situation or the response that we'd like to see in the patient? We need to be able to modulate the system. And by this, I mean two forms of modulation. Modulating the system itself. So how amenable is this system to modulation by genome editing, or by conditioning the cultures in which you're growing it in to represent the patient physiology, such as hypoxia or high glucose. And also modulation of the system by different perturbants. So we're seeing an increasing range now of entities available to us to modulate cells that goes beyond small molecules. So we have things like modified RNA, antisense oligos, inhibitory RNAs, viruses, antibodies, secreted proteins, and the CRISPR system. So how well does our model fit with these modalities to be able to affect its readout? Robustness. So we have, as we've touched on, a high need that it is accurate and consistent. And we need appropriate positive and negative controls so we know how well our system is working and that we are eliminating false positives and false negatives. And finally, cost. And this is a factor. We need a system that is affordable and that we can run in a screening context to the throughput and turnaround times needed. So considering some practicalities of working with 3D organoids. And here I'm thinking especially about readouts in the imaging space. So here there are several considerations and limitations we need to be aware of when using our organoids. So if you look in the top left-hand corner, you can see our 10K organoids. And these are organoids with 10,000 cells inside them. And these range from about 300 to 500 micrometers in diameter. Over on the top right-hand side, you can see some organoids you make which are 1K. So these have 1,000 cells inside them. And these have a diameter of around 150 to 300 micrometers. So when taking images of these organoids, it's tempting to think that you're looking through the middle, but using the technologies we have, we are quite limited to the depth at which we can image these organoids. And this is influenced by a number of factors, including the focal points of your reader, the depth at which your imaging agents can penetrate into the organoid, and the intensities at which they read back or which your imager can read, the deeper they penetrate into the organoid. And it's important to remember this when looking at the images, that these are cross-sections not through the middle of the organoid, but rather at the edge of the organoid, as shown in the top middle diagram. In the lower panel, we're seeing images that have come from different types of analysis of those images using our confocal. So we can use features such as max projection, so taking a number of stacked images and amalgamating them together to give an overall image of what we're seeing throughout the slice of the organoid that we were looking at. And in this first image in the bottom left-hand corner, you're seeing an amalgamated readout of the GFP fluorescence you can see following the nephron reporter. Alternatively, we can look at individual sections if you wish to look in more detail about what's happening at the different levels, and also follow features as they move through the organoid. We're constantly evolving and adapting our 3D organoid with drug discovery in mind. And here I've tried to list a few of these developments that we've made. So, sticking with the imaging theme, it's very important that our organoids are in a fixed position on the bottom of the plate. By that I described in the method, our organoids are generated in suspension. So we needed to develop methods that would stick our organoids down to the bottom of the imaging plate whilst retaining their 3D structure. In the bottom left-hand panel, you can see some bright field images of our kidney organoid stuck to the bottom of the plate. Much of the structure remains, although we do see some cell migration out from the organoid. But we are able to stick our organoids to a plate. The attachment is random, so we're not directing whereabouts in the plate that it's sticking down. And as such, when we're coming to image the plate or image the organoids, we want to make sure that the images we're capturing are the interesting ones and ones of the organoids rather than the empty spaces between. So here we have developed methods where we can do some searchable functions where the plate can be quickly read and identify where these organoids are. And importantly, remember which organoid is stuck where. And this is very good for when we're reading the plate again and again. And you can also come back at a later time point to re-image that plate, and you can look at the same organoid and see what's happened to that exact organoid over a time and also following treatment. So this has enabled us to generate an assay where we can rapidly image the points of interest within a plate in a 384-well plate format and a model at which you can image multiple times over the course of a treatment. And here, a final slide on some of the techniques we're using to validate our organoids and validate what we're doing. So I've spoken a lot about imaging, and you see in the top right-hand corner some images of sections through an organoid. And here we're showing features stained with LTL to show the tubular cells, stained with WT1 for our podocyte cells, and stained with CD31 in green to show endothelial cells. And if we look at the CD1 staining, you can see vessel-like structures forming, and you can see branches forming off these vessels. And you can also see the potential for a lumen to start forming. So seeing some nice features forming throughout our organoid. In addition to imaging, you can see how we're using immunohistochemistry on our organoids to look at structural features throughout. And from this, that you can see in the organoid that in the center, we're not getting a necrotic core. We're seeing cell features that we would see throughout the organoid. Next generation sequencing is also a powerful tool that we have applied to our organoids. This is both in the single cell space to see which cell types are within the organoid and what genes they're expressing, and also using bulk RNA-seq on the organoids as they develop over 0, 10, 20, 30, 40, and 50 days to see which genes are present and absent and when they go up and when they go down. And this is very useful when looking at our organoids, especially if you have a target in mind, because we can quickly check if the target is present, what level the RNA expression is, and at which time point it's increasing and decreasing. And all of these things are giving us confidence that we have a validated model. So now to move on to some examples of using these 3D kidney organoids. And the first example is looking at drug clearance. So here, we're thinking about looking at transport activity in renal models for drug clearance. And on the top, you can see a pictorial diagram of the kidney going down through to the proximal tubule cells, of which the project team is interested. And in there, we can see the transporters that the team are specifically interested in, notably OAT1, OAT3, and OCT2. So the team are interested in these because they are involved in the process of elimination of drugs from the basolateral membrane, removing them from the bloodstream. They do have models that exist today, and there's a number of cell sources for these, including immortalized cells like Hicks and MDCK, where you're overexpressing the transporter of interest, but that's not ideal for reasons I've mentioned earlier. They're also, you can use fresh or cryopreserved RP-TECs. I think these are seen as the gold standard for in vitro work, but they do have a higher physiological, I think, relevance than the immortalized lines, but their transport expression can quickly downregulate, and they do see batch-to-batch variation. So there's an opportunity here to apply our 3D organoids to validate this model. So the project team came to us wanting to see if our organoids would work in this system. And in the blue box is highlighted some of the requirements they put on us, and I've touched on these earlier. And so what we did is we first looked at the RNA expression data from our organoids, and this is shown in the bottom right-hand corner. So we can see an upregulation of the key genes that we're interested in our day 31 organoids as compared to our day 10. In the middle image, at the bottom, you can see the structures. So the interest is in the proximal tubule cells, and using LTL, we're staining for these, and we can see in the top right-hand corner of the image, a ring of these cells looks like they're forming a tubule with a lumen in the middle, and also flowing down, cell staining for E-cadherin, which could be an indication of forming further tubule structures like the distal tubule. So from initial looking at the data, it looked like we had the transporters expressed that we would like to see. So the experimental setup that we took was to take these day 31 organoids, and then in 2 different assays, look at either OCT2 or OAT1 or 3. And the way we would do that was to take a substrate that was FITSI labeled so we could image it, and then see how this was taken up into organoids in the presence or absence of inhibitors. And this ideally would be done in live imaging, so we could see the substrate being taken up, but there are a lot of limitations here. So we also saw if we would have to do fixed imaging, so where we would fix and permeabilize and then stain the organoid as well in the assay. So this slide shows some of the initial data on the OCT2 and OAT1 and 3 substrate uptake. So if we look at the top left-hand image, you can see when we add substrate to the organoids, they go bright green, indicating that the organoids have taken up the substrate. And in the bottom left-hand, where you see the same experiment but the presence of an inhibitor, there is much reduced uptake of the substrate into the organoids. And the same is true with the OAT1-3 specific substrate, again seeing good uptake into the organoids, and in the presence of an inhibitor, that is being inhibited. And on the right-hand side, you're seeing an untreated control where there's no substrate added, and you might be able to see a faint green background, and that's coming from the nephron GFP reporter. So from this, we could see we're getting specific uptake from the channels into the organoids, and it looked like it was going into specific compartments, but we couldn't tell from this imaging. So this slide, we're looking at a closer look at the organoids using both the green and the red channel. So in the top left-hand panel, this is the live imaging, where you can see proximal tubule staining. In the middle, you can see in the bright green where the substrate is going in, and with the dark green where the nephron GFP reporter is coming up. In the bottom panel, you see the fixed cells, or the fixed organoids. So in the bottom middle panel, you can see again where the LTL cells are stained. You cannot see any substrate because the cells have been fixed, permeabilized, and then got back in, and so that substrate is leaked out, but you can see where the GFP is expressing. So if you look at the image on the bottom right-hand side, we're seeing co-expression of the GFP reporter with the separate podocyte marker WT1. And in the top right-hand corner, we have a nice image whereby you can see red staining for proximal cells, and also where it's growing bright green, that is where the substrate is being taken up. The second slide shows much the same thing, whereby we're doing the same experiments but with OAT1-3. Again, you can see we're getting specific uptake of the dye into the organoids and into specific compartments. So if I just point you to the top right-hand corner, you can see substrate-specific uptake into the organoid, and it's being taken up by the proximal tubule cells rather than the podocyte cells. So to summarize, we're seeing uptake of the substrate in a specific manner. It can be inhibited by the specific inhibitors, and we can see it's being taken up by the correct cell type. So that's all positives. Unfortunately, quantification is very challenging to see how much dye is taken up, and also its uptake rather than transport across the lumen, so we can't do any next flux studies. But we do see this as a good start model for drug clearance. The second example I would like to give is looking at toxicity to see how PPAR agonists affect drug toxicity. So the data I'm showing here is from a poster presented at EUROTOX. And here, what we tried to do was unravel the toxicity induced by PPAR gamma agonists using our iPS-derived kidney organoids and transcriptomic analysis. So what we did here was to take our organoids and apply PPAR agonists, and then harvest after 6 hours, or dose again over 9 days, and then harvest after 9 days to see how the PPAR agonist had affected the kidney organoid. And the readout for that was transcriptomics. So from this, the preliminary PCR data showed us that the kidney organoid did demonstrate drug-induced gene expression changes, and these genes were associated with kidney toxicity, water reabsorption, and ion transport, as well as known PPAR gamma target genes. It was a little surprising, perhaps, that there were fewer significant changes in the transcriptomic analysis than we might have anticipated, but analysis is still ongoing. But interestingly, we did find a common gene target that was upregulated both by the classic PPAR agonist rosiglitazone and pioglitazone, and that's shown in the graph on the right. So to summarize here, although this work is early and analysis is still ongoing, we have identified a sodium amino acid transporter as a pituitative PPAR gamma target, and that's found to be expressed in the renal tubule cells. And we believe our data indicates that the iPS-derived kidney organoids may provide a platform to detect PPAR gamma-induced responses in a toxicity model. There are certainly opportunities to develop this model using things like single-cell analysis to be able to deconvolute which cell types are being affected by the toxicity. But ultimately, this is a good step forward in the identification of some safety biomarkers. So now, just moving towards some closing remarks. First, just to summarize things where we are with the 3D kidney organoid work. So for the 3D model itself, I believe we have demonstrated reproducibility of our model. We've adapted it to a drug discovery setting, allowing screening. We're seeing the emergence of 3D in vivo-like structures. And we've performed extensive characterization of these organoids, looking at function, phenotype, using imaging and omics data. And I think we've given some examples in the safety space of how we've applied these. We've also used these with CRISPR-modified iPS cells, looking at disease mutations and also looking at them for target identification and validation. I was not able to talk about these today, but look forward to talking about these perhaps at future presentations. We've also used them for biomarker discovery and in project screening. So as a summary for 3D organoids overall, I think we, and certainly many other groups external to AstraZeneca, have achieved a lot so far in this field, but there's still a lot left to do. Not least getting further data and evidence that the 3D models are indeed superior in their functionality and translatability compared to 2D models. There's lots of reasons of why they should be better, but I think more data to show that they are is needed. We certainly need to improve the scale up so that they could be applied for higher throughput screening. We also need to improve the culture, so not only to develop the organoids, but also the technologies that we're using to measure the outputs from these organoids. Our organoids are showing some key markers and some emerging structures, but they are far from mature. So work to further mature these organoids is certainly needed. One possibility would be to apply these organoids into microphysiological systems to introduce features like flow. So this should put things like shear stress onto the organoids and hopefully improve maturation, as well as giving us alternative functional readout. So this is all with a function on developing new medicine and also publishing great science. And with that, it just remains for me to thank the very many people involved in this work across AstraZeneca and our external collaborators, to thank the organisers once again for the opportunity to present to you today, and also to thank you for your attention. And I will now hand back to Roland. Thank you.
Brian, thank you so much for that very insightful presentation. We'll now begin the live Q&A portion of this webinar. So again, if you have a question you would like to ask, please do so by clicking on the Ask a Question drop-down box located on the far left of your presentation window. Type in your question that appears into the box and then click the Send button. Okay, so let's get started. We have a few minutes for questions. So we do have a number of questions that are in, and I'll start off here with Brian. From just a purely practical standpoint and based on your experience, what should newcomers to the field be aware of before stepping into the space of organoid culture? What basic advice can you give them?
I think going in with your eyes open, certainly it is culture every day, and it is hard work. So stem cells are not the easiest cells to work with, and they need a lot of attention. And then when moving into organoids, we are just complicating things immensely. So I think going with your eyes open in terms of the time it will take you. There are costs involved as well. It is not a cheap culture to do. And then also, I think you need to have the question in mind that you wish to answer. So what is it that you wish to answer, and what is it that the organoids will do that you are lacking at the moment? So I think you need to go with the key question in mind so that you can answer that with your organoid, I would say.
Very good. I have another question for you here. This is about actually generation of IPS themselves. And we have somebody here asking about the methodologies that you use for generating your IPS and why you have selected those specific methodologies.
So I guess there are a number of different ways to generate. I think this question is pointing at IPS from somatic cells. So there are a number of different methods to do that, and they're advancing all the time. I would say that our lab does that. I would also say that there are some very good labs and providers externally that do that very well as well. So we do some de novo generation of IPS lines, and we have a number of different methods to do that. They can be virus-based or mRNA-based, but typically we will look for an external provider to help us generate our IPS from a somatic cell.
Okay.
And there's a number of these. Sorry.
Sure.
Just to add, I think if you can do things, as they improve, I think if you can generate your IPS cells, I think if you can do that without integrating anything genomic into the DNA, that's a huge advantage. So it is the same. I think most of the technologies do that now, and keep them feeder-free as well.
Okay. Brian, another question for you here. There are many reports in the literature that IPS cultures derived from diseased patients may behave differently to their healthy control counterparts. And this particular user is wondering if you see differences in the ability of healthy and diseased IPS to generate your desired organoid types.
I think the answer is yes. I think when you're taking patient-derived cells, if you're just taking the native patient cells, then you can see variation, and I think some of that can come from the patient themselves. With the disease they have. A lot of it can come from the method that's used to generate those IPS cells. So I think to overcome that, I think patient-derived cells are very important, but I would certainly advocate the use of precise genome editing technologies to make isogenic pairs of those cells. And by that I mean, so either taking a native cell and mutating the gene of interest, or taking your patient cell and then mutating, if it's a mononuclear disease, that gene back. And I think there you have the perfect control because it's isogenic. So the only thing that's changed is the genes you've changed. I appreciate with a lot of diseases they're not monogenic, and so that's difficult to apply. But I think if you can get isogenic controls perfect, if you're going from patient-derived material, I think what you will need to do is you would need to have a number of cells from a patient. So both from the same patient, maybe of N of 3 from the same patient, and also from different patients to show that your results are reproducible across a number of lines from a number of patients. I think that's what you would need to do.
Okay, lovely. We have somebody who'd like to know if your organoids can be expanded indefinitely or do you need to continuously reinitiate them from your iPSCs?
So they cannot be grown indefinitely. So we can take them out. So we've shown them that we can culture them. So I guess they go into organoid formation on day 12. We've taken them to day 50. You do see changes over that time course, and you do see maturation, but it's not indefinite culture. You will see deterioration after a while. And so we have routine generation. So every couple of weeks, we will start a new round of generating these organoids.
Okay. From the drug discovery work that you've shown, I wondered if there's any evidence to date that shows that your organoid work is actually generating higher quality candidate drugs.
I think that's too early to say. I think for the reasons I've given, I can understand why it would be more predictive. I think you see better functionality, better prediction. I think, unfortunately, because of the length of time the drug discovery process takes, so to going from developing a molecule, taking it through discovery, development, and then into the clinic, I don't believe we've seen data yet whereby a molecule has been developed and discovered in a 3D system and gone into man. So I think there is no data there yet that would confirm that, but it would be my belief that we will see that eventually.
Some of the work that you describe, Ryan, involved the use of organoids to look at parameters such as drug clearance. How familiar are the regulators with organoid technology, and do they accept experimental data derived using organoids to be substituted for classical tests, or is this simply an additional layer of data that's being provided?
Unfortunately, I can't speak for the regulators, I guess, on their level of awareness. My belief, talking to our discovery, safety, metabolism colleagues, is that I do believe they are becoming more aware. I do believe, certainly in the safety space, we are seeing data that is showing better prediction from 3D organoids. And this can be in the kidney space with drug clearance, but also in the cardiac space with cardiac organoids. I think there is a growing body of evidence that from an in vitro prediction point of view, we are seeing better prediction when you move from 2D to 3D. So I believe they are aware of it. How close they are to acceptance or substitution is a different question. And then I guess ultimately what you would like to see is the increased use of these in vitro models and a reduced use of in vivo models. But again, you'd have to prove that these are at least as predictive as other models before doing that. So I think it's possibly an extra layer at the moment, but I think the ultimate aim is it is a reduced layer and a reduced number of assays and a reduced amount of in vivo work.
Okay. We have several questions actually, Ryan, which I guess are heading towards the cell therapy space. You've described essentially the use of organoids in the drug discovery workflow, but we have several people here wondering if AstraZeneca has ambitions to use organoids or cells as the cell therapy itself.
What I can comment on is that from within AstraZeneca, we do have an interest in stem cell therapy. I think if you take a look at our website, you can see our press releases on that, and we have certainly programs in the cardiac space using stem cells as a cell therapy. So what I would say is that we definitely do have an interest in stem cell therapy. The application of organoids is an interesting question, but not one I can answer at the moment.
Okay. You've touched at the beginning of your presentation on a number of different organoid types. We have one user here who's interested to know if there are 3D skin organoids.
I'd have to apologize. I'm not sure if there are. Skin is not a relevant area for us, so I don't have much focus there. So I wouldn't be able to answer that accurately. I apologize.
No, I think there are, since a number of years, I think there are some skin models which require liquid air interfaces actually for culture.
Yeah, certainly a number of models, and I guess for wound healing, I guess you see that, and I think in the 3D printing space, I think you see quite a lot of skin models. So I think certainly not only for the use in pharmaceutical, but I think the cosmetic area uses these a lot as well. So whether it's an organoid, but certainly some 3D models in the skin I'm aware of, but I'm not an expert in that area.
Okay. I think we have time for one more question, actually, Ryan. So it's a fairly simple question here, but we have a novice who'd like to understand how large these organoids can actually get.
They can get pretty big. We can make organoids of different sizes. So in the publication, I've talked about organoids of 1,000 cells. In the publication, we have organoids there of 100,000 cells. At 100,000 cells, we're still not seeing necrosis in the middle. So for the organoids, how we culture them, they're looking nice. There will be a finite point, I think, whereby you're stopping nutrients getting to the center. That may be of benefit if you're thinking about a cancer sphere or a cancer organoid when you want to see hypoxia in the middle. So I think there will be some limiting factors about how big your organoid can go, and they may be good or bad features. We have taken ours to 100,000 in size, and they look okay. I presume you can go bigger, but we haven't looked at that.
Okay, lovely. Ryan, thanks very much again for the presentation and for taking so many questions. And I'd also like to thank the audience for taking the time to listen in. Before we go, please note that today's webinar will be available for on-demand viewing through to February 2009. And thanks very much again for everybody for joining us today, and we hope that you enjoy the rest of today's event. So as of now, this presentation will close. Thank you very much indeed.